Hello, welcome to my channel. Today we'll discuss about study of bacterial pathogens. Study of bacterial pathogens with respect to classification and biochemical characteristics, antigenic structure, viability characteristics, pathogenicity, pathogenesis, symptoms, laboratory diagnosis, epidemiology, prophylaxis, and the chemotherapy. We will study about each pathogen. Bacterial pathogens, in which two pathogens we will study. First one is E. coli and second one is Staphylococcus aureus. In which first we will study Escherichia coli means E. coli is bacteria that is found in intestine of the both humans and animals. In most of the cases, bacteria is harmless and helps in the digestion of the food. Certain strains of the E. coli can cause the infection and the symptoms, including the diarrhea. Some of the infections can be dangerous. This genus is named after Escherichia, who was the first to describe the colon bacillus. E. coli normally lives in the human intestinal tract. But it can cause the serious infection. Gets into the urinary tract. Most of the E. coli strains causes infection of the bladder, that is the cystitis. This type of the urinary tract infection is usually occurs in the woman as a result of the sexual intercourse. Classification: most of the strains of the E. coli form lactose rapidly and they produce the indole. There are the 150 different or the distinct O antigen and the large number of the K and the H antigen. Bacterial characteristics: main biochemical characteristics: ferment the lactose. E. coli is capable of producing the ethanol, acetate, and the carbon dioxide. Most of the strains are the positive for catalase but negative for oxidase citrate urease. They shows the indole production and methyl red taste. Next one is antigenic structure. E. coli strain contains the four type of the antigen. For first one is the flagell antigen, that is the H antigen. Second one is the somatic antigen, that is the O antigen. Third one is the surface antigen, that is the K antigen. And fourth one is the febrile antigen. Next one is the viability characteristics of the E. coli. E. coli is a gram-negative rod-shaped bacterium, is about 0.6 micrometer in the diameter and the 2 micrometer in the length. Bacteria from the non-splitting colonies shows characteristics green sheen in the EMB agar. E. coli shows the peritrichous flagella. Some strains also shows capsule and the fumary. Next one is the pathogenicity of the E. coli. E. coli shows the two types of the virulence factors. First one is the surface antigen and second one is the toxin. Surface antigen, that is the O. E. coli shows the endotoxic activity and protect the bacillus from the phagocytosis. The envelope of the K antigen protect the bacterium from phagocytosis. Most of the strains of the E. coli are responsible for neonatal meningitis and the septicemia. Fimbric causes the UTI, that is the urinary tract infection. E. coli produces the exotoxins and the enterotoxins. There are the three distinct enterotoxins have been identified in the E. coli. First one is heat labile toxin, second one is heat stable toxin, and third one is the virotoxin. Next one is the pathogenesis of E. coli. Most of the E. coli produces the UTI, that is the urinary tract infection. The ability of E. coli to produce the UTI is related to the general virulence. Factors such as the hemolysin together with the pili mediated. Relatively minor injury of the mechanical effect of the sexual intercourse have been shown to allow the bacteria access to the bladder. Bacteria bind to the urothelial cells via the P. pili. This attachment allows the bacterial access to the bladder. This blinding causes the disintegration of the bladder. Next one is the diagram of the pathogenesis. That is the next one is pathogenesis of the urinary tract infection. Next one is the symptoms of the E. coli disease. Most common symptoms of the urinary tract infection are the diarrhea and the urinary frequency. Infection produces the pyelonephritis, fever, flank pain. Infection usually begin with the mild watery diarrhea. The duration of the diarrhea is different for the different strains of the E. coli. Next one is the laboratory diagnosis. E. coli is rapidly isolated in the culture. E. coli strain commonly found in the stool presents in the special problem. Immunoassay and the nucleic acid methods have been described that they able to detect the toxins and the gates associated with the virulence. Next one is epidemiology of the E. coli. E. coli occurs in the more than 7 million cases in the cities in the 250,000 of the piloparititis estimated in occur in the healthy individuals every year in the United States. Urinary tract infection more common in the human. The reservoir of this infection is the patient's own essential intestinal E. coli flora, which contaminate to the perineal and the urethral area. Next is the prophylaxis. Avoiding uncertain water, ice, salad, and the raw vegetables is a wise precaution which traveling in developing countries. And last one is the treatment of the urinary tract infection. Because most E. coli diarrhea are mild and the self-limiting, treatment is usually not an issue. Rehydration and the supportive measures of the meningitis therapy treatment with the tenanthorium. So